Okay, I'd say we can go ahead and get started.
Perfect. Alrighty. Well, thanks for having me. It's my first ever talk outside of the department, so I'm pretty excited for that. Today I'll be talking about not just EPR and how we can utilize EPR in various different ways, but I'm going to be applying EPR studies to analyze the impact of metal organic framework crystallinity on orientation of enzymes and the dynamics of enzymes when we use them to biomineralize the protein. I'll give a slight warning and precaution. I'm not normally a EPR scientist. This is my first ever project with EPR, so I'm going to be focusing pretty heavily on how we can apply EPR to applications and hopefully get these materials off the ground. So it's more focused on how we can use EPR to characterize these materials. Okay, no, it's not. Hold on, give me a second. It's not clicking. There we go. So just to start, I'm going to talk about what is a metal organic framework, or also known as a MOF. At the very basis level and foundation, we have organic linkers, which could be anything from 1 or 2 or 5 benzene rings that usually have a carboxylic acid on each end, and then a metal ion. And these vary depending on whatever MOF you're trying to analyze or use. Next, at the secondary level, we have the secondary binding unit, which is usually our crystal cell. As they start to build up these frameworks, they have interconnections between nodes. And then from there, you get ligand bridging to form pores, as you can see in the middle of the screen there. There's blank space that forms pores to create an inner framework before we finally get to a overall morphology on the micrometer level with coordinatively unsaturated sites, where at the surface sometimes metal ions aren't fully saturated, so they can be further modified post-synthesis to do things such as modify or attaching drug molecules that can be reversibly bound and delivered to targets. How we synthesize these these materials, there's a lot of different methods, and I only show 5 on the right here. There's probably closer to 10 or 20, but these are some of the more common ones. And in my lab specifically, I use hydro/soluble thermal methods for high temperature, high pressure synthesis of metal organic frameworks in a PTFE reaction chamber. These form very highly ordered metal organic frameworks that have high crystallinity, high stability, which can be very desirable depending on the application of the material. Or there's also co-crystallization, which is essentially crashing out or precipitating out the MOF from solution. So this is a very green and benign synthesis. It's just a water solution of metal with a water solution of ligand that we combine together to form these metal organic frameworks and then wash them with water. So it's very green, very minimal waste. The drawbacks, however, are the lower crystallinity compared to soluble thermal methods, and that can sometimes actually be used as an asset more than drawback. So we'll get into that in the applications. And then something I've been looking into recently is mechanosynthetic methods, where we use ball milling methods, pretty much just steel ball bearings rolling around with the starting materials. And it almost bridges the two, where we can get increased stability and crystallinity while not impacting the green synthesis that much of co-crystallization. So it's kind of the mix of the two to get the best of both worlds. So why do we care? The importance of MOFs is that they can be used in very many different things. I've included about 7 different uses here. However, there's a ton of other ones that I couldn't include because the slide would be way too long. Some of the important uses of metal organic frameworks include drug delivery, so that you can get to a cancer cell or maybe get to a target where you have more acidic or basic conditions. And then upon the MOF arriving there, the slight pH change could either deprotonate or protonate the drug molecule and allow for the delivery of that drug to the target. There's gas storage. We all hopefully know that Many gases such as hydrogen are very flammable, very dangerous to ship and move around the world. So if we can adhere the gas to the metal organic framework reversibly, we can attach it to the MOF, store it or transport it safely, and then reversibly detach it from that material. Some of these materials are also catalytically active for a wide variety of different reactions, and I've actually shown in a publication we're starting to write up right now that one of our protein assays is actually catalytically enhanced by empty MOF even without the protein present. And waste treatment, these materials have very high surface area. If you look on the right, you can see there's pores between the material where the ligands and the metals don't have, or there's empty space. And this empty volume leads to very high specific surface area or VET area, which allows for them to uptake any ions or organic pollutants with high efficiency. And if you combine the two, there's actually been a few studies to show that we can manage pollution through catalytic degradation using the metal organic framework and then binding the products from that to the metal organic framework to treat waste. You can also use these as a separation to clean up impurities from reactions. I've underlined protein immobilization here because this is what we're focusing on with our studies and what we'll be getting into very shortly. And you can also do stuff like ion detection. If you're hoping that there's no chloride ion, for example, in your solution, you can have a MOF that changes colors once it's in the presence of chloride, and you'll see that detection. So as I said, we're going to be talking about protein immobilization, and why this is important is proteins are very important for all biocatalytic activities. Most of the reactions biologically in our bodies happen with the aid of an enzyme. These enzymes help to speed up reactions almost or more than 1 million times in some cases. And the one drawback of enzymes and why we can't use them commercially for a lot of things is they're very unstable. One of the ways to get around this and stabilize these proteins is to attach them to some sort of support material or a support framework. And through this attachment, they're stabilized so that they can be used and utilized multiple times and in harsh environments, such as in environments that have organic solvents or elevated temperature and things along those nature. I show only 3 methods here, however, there are more. On the left, we have physical absorption of an enzyme onto a support. This is usually reversible and can be used for biocatalytic reactions once the enzyme is no longer working, or if you want to recover the enzyme, you can reversibly remove it from the support. In the middle, we have polymeric materials or porous materials. You can encapsulate proteins either fully or partially in polymeric or porous materials such as MOFs. And this helps to stabilize them greatly. However, it does impact the catalytic efficiency of the enzyme. So you kind of have the trade-off of it's less efficient, but since it is more stable, you can use it for more cycles, and therefore it's still pretty green. And then lastly, you can actually bond the proteins directly to a support through binding to outer residues of proteins such as lysine, cysteine, aspartic acid, and glutamic acid. In a paper recently published in my group on the right, you can see that depending on where, if we use site-directed spin labeling EPR with the MTSL spin label, we can actually analyze the confinement of these proteins based off of EPR signal. If a MTSL spin label is exposed at the top of, say, a metal organic framework, and it is free to move around, we will see a mobile peak on the right there. Whereas if the protein is buried deep within the MOF, there may or may not be mobility depending on if the confinement is very packed and there's not a lot of room for the MTSL spin label to move, or if it's completely immobilized. So this is where we've started our EPR studies and analyzing these metal organic frameworks. Before I get fully started, I want to say just before I joined this group, there was a paper done by the students in this group with expanding the 10 MOF library, it's important to be able to characterize many different metal organic frameworks, and there are thousands out there. However, there's not a great system for seeing exactly what metal organic frameworks have what properties. So this paper kind of aimed to synthesize 10 MOFs with co-crystallization, that green synthesis method, to show the characteristics such as XRD, crystallinity, things like that. So the work that I'll be presenting today builds on this work of 10 MOFs that utilize zinc, aluminum, copper, zirconium, or nickel with either BDC or BPDC ligand. That will be in coming slides what that means, but it's benzene dicarboxylic acid or biphenyl dicarboxylic acid. And the project goals that we had for this project was to, as I said, expand on this library of 10 MOFs to show enzyme orientation and dynamics when we co-crystallize the metal organic frameworks around the proteins, and see is there any activity correlations in this work? Can we see any noticeable correlations with the EPR data and with the physical properties of the metal organic frameworks? So this is the paper that we recently published this year. As I said, we utilize the MTSL spin label. The model protein that we use is lysozyme because it's very stable. It's very easy to work with. And so when you have a chemist like myself who doesn't really work with proteins often, I don't absolutely denature my protein before we even get things started. We labeled 8 spin labeled sites between 44 and the one that's not on this diagram because it's in the back of the protein there is 151 with the MTSL nitroxide spin label. And from there, we can see based off confinement in the metal organic frameworks, the mobility of the spin label and deduce the dynamics and orientation of the proteins in the materials. So how we interpret the EPR data is, as I just said, depending on if the spin label is free to move at the surface, say we only have partially immobilized and partially encapsulated protein. If the spin label is exposed above the surface, we will see that increased mobility. Or if it's buried in the lattice, the crystalline lattice of the MOF, we should see immobility in the spin label. And using EPR spectral simulations, we can deconvolute all of these components. So since this was my first ever EPR project, there was a lot of learning to be done here. And we have 64 spectra since we have 8 different spin labels and we have 8 different MOFs. You might ask yourself why I don't have 10 different MOFs, considering we're expanding on the previous paper of the 10 MOF library. The issue with that was copper-2 is EPR active, and after I tried removing the baseline of copper MOF without protein from the protein and MOF combination, for most of them, it was just too much copper that once we removed and baselined out the, or blanked out the copper, we actually didn't see any MTSL spin label peaks. And this is likely due to the high concentration of copper compared to the very low concentration of 1 MTSL spin label per protein, and we're not getting a ton of protein compared to copper in these solutions. I also had to baseline these spectra because if you see on the left here, the black spectra is not quite zeroed. There's quite a bit of deviation from the 0 mark on both sides of the spectrum. So I started off with manual baseline correction in Excel, and this got us pretty close to being a baseline that's zeroed before I would then put it into a baseline correction software and narrow it in even further with that. From there, we would normalize the area under the peaks and center the center peak in our spectra before we would do spectral simulations. And you can see the raw data on the right here for the 8 MOFs that we chose. All right, so for this simulations results and conclusions, what we have found was there's 3 sample spectra on the top here. We would have our raw data, as you can see, and we would try to match the spectral simulation as close as we could within reason. And then from there, we could use the spectral simulation software to deduce the immobile and the mobile components of the spectra and from there, we would have all of the the spectra constants that come along with spectral simulation, such as the rates of the immobile and mobile peaks. So what we generally found is that the restriction of our protein increases with crystallinity, which isn't too surprising. Our hypothesis was if we had highly crystalline materials, we would expect that the MTSL spin label and protein would be buried deep within the material and would be unable to move, and so they would have higher immobility peaks. There are components compared to that of the mobile components. And we saw that in our data. And we also found that SDSL then can therefore almost indicate MOF crystallinity and material crystallinity for protein immobilization, which is an interesting tool because I don't know, some universities might have less capability of using XRD and crystallinity characterization tools, or at, like our university, there's only 3 or 4 students that are allowed to use the XRD. So being able to use something different, such as EPR, we can utilize this as a tool for almost indicating, is it high crystallinity or low? And we also found that BPDC MOFs had higher restrictions compared to the BDC MOFs, which is interesting because the biphenyl MOFs, we would expect to have larger pores. So with the larger pores, we'd expect that there would be more mobility of the spin label. However, we didn't actually see this when we performed our scans and our tests, and we tested multiple times just to make sure that this was actually correct. And what we actually ended up deducing was from the previous material done by Jordal and colleagues before I joined the group, we found that BPDC MOFs that we form with co-crystalline reactions actually aren't metal organic frameworks in the sense that they're highly ordered 3D materials. They're more closely related, or should be called, metal organic materials. And the reason this is because they form 2D sheets of long chains of metal of the ligand and the metal in repeated order, instead of the 3D network we would expect for metal organic frameworks. So what how we rationalized these results was that with the spin label being more confined and more restricted in these BPDC MOFs, we think what was happening was that the 2D sheets would pack together very tightly and actually increase the confinement and rigidity of the protein immobilization. So it's actually less likely to be able to move around. So we tried to correlate this with biocatalytic activity. So from our previous publication, we know that the activity of our proteins when we confine them in these materials. And what we found was pretty interesting. High crystallinity MOFs, the activity highly depends on where the site or the active site is likely to be in the MOF. So on the right here, you can see kind of the orientation that we expect based off of our spectral simulations and what spin labels were most likely to be mobile and compared to the spin labels that were like to be immobile. You might notice that on the top right, zinc BDC actually doesn't show any orientation. And this was because there was no spin label that was more likely to be mobile versus immobile when you compared the 8 spin labels all together. So we concluded that there was completely random orientation for all proteins, and there was no almost preferred orientation. It should be noted that these orientations are educated guesses on the most likely orientation based off of which spin labels on the protein were showed to have the highest mobility. It's not going to be exactly the same for every protein in the material. And we found that for gel-like or amorphous materials, or even our multi-phase crystals, where there's various crystalline structures in our metal organic material or framework, these had higher activity. Didn't matter if there was the active site exposed or buried. If on the bottom right, if you look at the aluminum BDC and BPDC, the activity is phenomenal compared to, say, the zinc BPDC on the top, even though the active site for the aluminum BDC MOF is likely to be buried within the metal organic framework or just barely be poking out for the aluminum BPDC. What this means to us is since these gel-like crystals that are very amorphous and we don't see high crystallinity with XRD, have high activity, that they're likely to be flexible and allow for proteins to move and bend and be able to utilize that active site for this large substrate, since these activity tests are done on cell walls, is what lysozyme breaks down. So it's large substrate, and these MOFs allow the cell wall to get into the active site regardless of where it is in the material, so that the optical density of the cell wall goes down over time relatively quick compared to other materials. Some future directions that I'd like to take this, especially with EPR, is to test defect modulation in these materials. It's been shown in other studies that increased defects increases catalytic activity of metal organic materials or frameworks. And the reason why I think that we could use EPR here is when we were doing our EPR tests, we found something very interesting and peculiar, which is on the right here, and I'll explain that. We have a blue and a black line. It might be purple, I apologize, I'm colorblind. But what we saw is, since we had a spin label, specifically spin label 44 and spin label 109, those proteins, they didn't really act very well in the fridge. So we would express the protein, the biochemists in our lab would, and then within a week or two, we would get aggregation of the protein. So we had to use our protein very quickly, or make more every time we needed it, which was more of a hassle than we'd like. So what we tried to do is we tried to use a lysis buffer that contains 0.05 molar EDTA. So a very dilute solution of EDTA is in this buffer to stabilize the protein and hopefully reduce aggregation. And what we found when we did MOF synthesis and biomineralization with these proteins is that we had completely different EPR spectra from what we were originally observing. And that was kind of interesting to us because we didn't expect that at first. So we had to dive into what was going on there. And we found that if you have EDTA in the solution, the mobility of the spin labels is actually increased. And this makes sense because EDTA chelates to metals. So if we were trying to synthesize metal organic frameworks with EDTA in solution, what could be happening is the EDTA will actually bind to the metal, not allowing for it to participate in the formation of these metal organic frameworks. Or what could also be happening is once the metal organic framework is synthesized, the EDTA can bump into it and extract the metal ion from the metal organic framework, forming a defect. We can't say for certain which thing is happening yet because we haven't done post-synthetic addition of EDTA yet, but those studies will be done fairly soon here to see can we do post-synthesis modulation of these materials, and which would increase the activity through the addition and the increased mobility of our proteins. So we'll be testing those studies. Next, I also would like to test the metal organic framework particle size. Depending on how we analyze or how we synthesize these materials, the protein encapsulation characterization could be different. We might see completely different EPR studies and other activity studies, depending on exactly how we synthesize these MOFs, whether they're in dilute solutions or concentrated solutions, or if we use solvable thermal or mechanosynthetic solutions. So we would spin label and do our best to hopefully keep the protein alive while encapsulating it, and test the dynamics and orientations with the varying synthetic methods. And also in another paper that it's currently in preparation and almost out, we've shown that some of the metal organic frameworks in our studies that we work with are actually photoactive. So when we shine a light on them, they electrochemically induce a current in the solution. And this is likely due to the ligands capturing the light and then shuttling a electron from the ligand to the metal center, almost like an LMCT reaction in inorganic chemistry. I'm hoping to be able to utilize these MOFs with redox proteins and then study them with site-directed spin labeling and EPR to find exactly how they're being encapsulated in these materials. So I know there wasn't a ton of EPR in here, probably compared to most EPR seminars given in these talks. However, I think it's important to show how we can utilize these EPR spectra and simulations for applications in materials, and hopefully be able to show that EPR can be used for deciding what material we want to use when we're trying to find when we have an application and we're trying to choose which material we want to use. So, for example, if we're trying to do drug delivery, maybe we want a co-crystallized MOF that can break down once it's reached the cancer cell, but also then we can analyze how that material would act or how it would encapsulate these proteins utilizing EPR. So with that, I'd like to give a few acknowledgements to my PI, Dr. Zhangyu Yang, and everything he's taught me, especially with EPR over the last year. I would like to thank Zoe Armstrong, who was actually co-author in this paper. She did have the work with me, and we worked pretty closely together. Since this was kind of the first time I used EPR, I'm very grateful for her help. Maggie Leonards did all the protein expression in our lab, along with Allison, our undergraduate student, so I'm grateful for them. And then Chavin Lee helped to introduce me to spectral simulation and how I could acquire all of the spectral parameters from our raw data. Also, I'd like to thank Kelly Johnson for helping us create some of these metal organic frameworks when we didn't have the time, and our high school student Patrick Shen over the summer of 2023 actually took my Excel sheet for baselining our spectra and made a interesting program that expedites the process, and it's very helpful. So without further ado, are there any questions?
Oh, yeah, thanks, Austin, for that fantastic talk. That's some super interesting results you have there. First, we can try and take some questions from the audience. You can either unmute yourself and ask directly, or you can type it in the chat.
Oh, yeah. Sorry.
If no one else has some questions, I have a few myself. On one of your slides, you mentioned that the newer MOFs that you're synthesizing have higher restriction, but and I'm completely naive when I ask this, doesn't that make a somewhat, I guess, restricted environment if you're trying to do interesting chemistry with the enzymes that you're covalently attaching there?
Yes, actually, so maybe I didn't fully explain this that well. So part of these MOFs,
or I just missed it.
That's completely fair. Part of these MOFs is with crystallization, which is we're trying to do green synthesis of these materials. One of the downfalls is they have less crystallinity and therefore less stability. So the idea is we could use them for some applications. You might want high stability and larger pores so that the enzymes do have that increased activity. However, these frameworks could stabilize the protein until it's in an environment that we want it to actually maybe release the protein, and so that it can do catalytic activity in the solution then. Also, depending on the confinement of the protein, it actually could be more or less stable based off the framework stability. So really, it depends on what your application is. If you're working in organic solvents that are pretty harsh for proteins and would normally denature a protein, maybe we want a more stable MOF to keep it from unfolding and denaturing in that solution, in which case the confinement could actually help to make sure that it stays its active conformation.
Ah, okay. That makes much more sense. And then I suppose in that case, restriction could be quite beneficial. Is there a correlation with what you're referring to as crystallinity here and pore size?
So the pore sizes really vary. As I mentioned, where was it? On this slide, since these materials are more metal organic materials and not conventional MOFs with high porosity, or like with these giant pores between the metal nodes and that are created from the ligands kind of spreading everything out. It's very hard to tell. The SEM imaging doesn't show us a ton here. And then on top of that, when we did do BET in the previous publication in 2022, we didn't find these materials to have great high specific surface area. And we attributed this to potentially pores collapsing because of the less crystallinity and maybe less stable materials when we dry them out for BET analysis, or maybe since we also thought that they could be 2D sheets for some of these materials. Let me see if I have a picture of it here. Kind of like, I don't think I have a great picture of it, but if they're 2D sheets that are just stacking on top of each other in layers, almost like a layered double oxide or hydroxide, which is another material that does layered or has layered morphology and structure to it, we might not actually see very great porosity. So it's hard for us to analyze the exact pore size for these materials, if that makes sense.
Sure. No, that makes perfect sense. I mean, these are highly variable materials from what I understand from your presentation.
Yeah, depending on a lot of synthetic parameters, you can tune your materials to exactly what you need, which is also a drawback because then there's thousands of metal organic frameworks in literature.
Yeah.
And it's very hard to sift through the exact material characteristics that you might want for your application, which is why we've been trying to almost make like a library of characterizing different MOFs to make it easier pretty much to see how or what these characteristics are, and maybe you'll want to use this one versus another one, if that makes sense.
Yeah, sure. Just to be able to dial in what you need and pick one off the shelf.
Yep.
I'll open the floor to the audience again before I ask another question. Andreas, you're welcome to unmute.
Hi. So can you hear me? All right?
Yes.
Good. So I have a technical question concerning the baseline correction. So on slide 11, you showed that on the left-hand side you are above 0, and on the right-hand side you are below 0. So did you just correct it empirically so that you end up on 0 on both sides, or do you have any like a physical model or so?
So the easiest way we found to do this was we had a EPR spectrum of our blank, so it would just be water with our EPR tube and any signal from the cavity as well, and we would subtract that from the baseline or the raw data that we collected from the EPR, and we had to manually adjust a baselining factor because when we did a high amount of scans, our EPR would spit out baselines that had much higher intensity than the actual spectra themselves. So we had to apply a baseline correction factor as well to that. And so I would manually change the number until the spectrum was as close to 0 as possible on both sides. From there, we would take that raw output because humans are imperfect, and we would input that into a baseline correction software that was developed, I believe, by Altenbach and Hubble a while ago.
Okay, thanks.
Yep.
Any other questions from the audience? Yeah, I'd like to ask another question, if that's all right. So you sparked my curiosity here. What exactly is a defect in a metal organic framework?
Yes, so a defect would almost be, let me get back to a easier. So in this picture on the right here, this is actually known as MOF 5. So when you solval thermally make zinc oxide metal cluster, form a MOF with BDC linker, what you get is this highly structured 3D MOF, and we only show 2 dimensions here, but you can see the pores in the middle here. Defects and defect modulation would be almost on a morphological level where sometimes these linkers or even the metal would be removed. Maybe there wasn't enough metals on the surface of the metal organic framework when we turned off the reaction, or maybe it used up all the starting material. It would just stop and not have the metal there, or wouldn't have a linker between some metals. It doesn't have to be exactly ordered like these materials. And the defects have been shown to increase catalytic activity in some metal organic frameworks. The one that specifically comes to mind is named UIO66. It's very popular MOF, probably one of the most famous, and it's used for many different applications because it's very stable in most environments.
Okay. Would that so could you then go back and utilize the EPR active MOFs to sort of interrogate those defects?
Yes, so kind of like we did on here, where we saw a difference in the MOFs, or specifically we saw a difference in encapsulation of our proteins with metal organic frameworks here. There was a lot more mobility in our spin labels, and we just chose a random spin label, 89 here, for this method. But when we did other tests with other spin labels, we saw a similar trend. We can almost see what the mobility is and like how the defects can be modulated with, say, EDTA, or there's other ways of causing defects in MOFs, such as adding dilute acids or bases and things like that. So we can almost probe that with EPR, utilizing a spin label in encapsulated protein.
Okay, I was thinking more of going back to the copper MOF you were speaking of earlier. I think that would be kind of cool.
Yes,
and I'm just purely speculating, but
we've tried to do, and I actually have some data for copper MOFs for CWEPR. It's very hard for us to collect, but and we haven't really seen great data that's conclusive of anything currently. I haven't tested it with EDTA, however, so that would be a future avenue to test. We've also tried doing deer EPR with, you can make mixed metal MOFs, so maybe you have a ratio of one metal than the other, and we were hoping to see, is there almost like set distances between the copper ions or copper atoms in the metal organic framework. However, we didn't get any conclusive results, and we don't actually have the capability of doing deer EPR at our university, so we have to send out the samples, so it takes quite a bit of time. It would be a very interesting avenue to explore, though, with those methods done.
Yeah, definitely. Just sparked curiosity. Okay, one last call for questions from the audience before we close. I don't see any hand raising, so all right. I hope everyone will join me in thanking Austin for such a nice presentation. And with that, I will close the meeting and hopefully see you all again in 2 weeks. Thanks again, and everyone have a nice day.